Hello everyone, welcome back to another video. Today I'm going to be covering PCR. I'm going to be covering a lot of things PCR. Um, so yeah, let's get started. I have a game plan as always, and I'm going to tell you what we're going to be talking about today. Okay, so for today's game plan, we're going to go into like a little bit of a background for PCR, the uses of it, what you need for PCR, and we're going to be talking about primer design, making sure things are in frame, melting and annealing temperature, restriction sites and cloning vectors, and quantitative PCR, as well as reverse transcriptase or RT-PCR. So yeah, let's get started with a little bit of a background of PCR, what it is, and what it does. Okay, so what is PCR? PCR is a process that amplifies small amounts of DNA by successive rounds of application. And you may ask, what does that mean? Basically, you are replicating this certain amount of DNA, so like a gene of interest or like a certain sequence that you really want to look at, you're going to be amplifying those DNA and just you're basically replicating it the whole time. And so what is it used for? It's used for DNA fingerprinting, so like in criminal cases, genetic history, so evolutionary wise, like looking at um, your DNA from your ancestors or from other organisms back in time, as well as COVID tests and gene fusions, so DNA sequencing. Okay, so the next thing is going to be requirements of PCR. And so PCR requires a DNA template. And so a template is required because remember when you learned about DNA replication, you need a template. So you need a parent strand, and that parent strand is able to be used as a template for the new strand or the daughter strand. And so the next thing you're going to need is DNA primers. And these primers come with their own melting temperatures, but together they have an annealing temperature, and I'm going to talk about that a little bit later. And then you need your nucleotides, so they're DNTPs. And I feel like I did talk about this in the carbon metabolism video. So if you haven't watched that, go watch that. And you also need DNA polymerase or just a stable polymerase. And so I don't know the exact DNA polymerase that's going to be used, but for a general sense, just think of it as just the general DNA polymerase. So yeah, let's get into PCR, kind of just a general sense. Okay, so for PCR, you're going to have your DNA. And so DNA comes in two strands, right? So because we want to be taking a parent strand and then adding a primer onto it to create a new strand, we are going to have to split the DNA. And to do that, we're going to have to break it up. And so by doing this, we just have to heat it up to denature the DNA, and that gives us two separate strands. So now that we have split up our DNA, we are going to be able to work on them separately. And so the gene of interest here is in blue. I hope you can see that. I know it's more visible towards the end because I really did color it in on the board. But at the top, I kind of put an arrow towards it, so I hope you can see that. But you have two strands. So one of them goes 5' to 3'. The other one goes 5' to 3' in the bottom strand, but the opposite direction. So now at this point, you're going to add your primers to both strands. And we're going to talk about what those primers are and how we make them in just a little bit. But we're going to take our primers and we're going to add them to both strands. We're going to anneal them. And so we have an annealing temperature. And we're going to be talking about that in a little bit. But both strands, you're going to have a primer annealed to them. The forward primer goes on the bottom strand. So the one that starts with 3', that strand is going to have the primer attached to the gene of interest, okay? And that primer is going to go 5' to 3', but it's just the forward primer is on the bottom strand. The top strand has the reverse primer. That top strand goes from 5' to 3', and we're going to attach the primer to our sequence or our gene of interest from 5' to 3', so it's going to go the reverse way, okay? So now that we have that settled, we're going to elongate those primers by DNA polymerase. So we're going to be adding our nucleotides with DNA polymerase. That's where our DNTPs come in, and that's where our DNA polymerase comes in. So now that our primers have extended, you can see that they hang off a little bit from the our gene of interest. And I don't know if you can see that on the photo, but I'm going to be doing like a little video of a close-up in the next minute or so, and I'll be showing you kind of more like I'm just going to review it over again so that you can understand the subject. And you're going to be seeing those little ends that come off from the gene of interest. So if you can't see it on the photo, it's okay. But right now, we're on step 4 on the board. Actually, we just finished step 4 because we did talk about how DNA polymerase elongated the primers with DNTPs. And so now that we have our amplified gene of interest, we're going to be separating strand, like the primer strand, or not the primer, the primer elongated strand and our gene of interest strand. So our OG strand and our red strand from both sides. So I don't know if you can tell, but I have little arrows there that kind of look like it's supposed to separate. So we're going to be amplifying those 4. So now we have 1, 2, 3, 4. As in the blue one, the blue black one is 1, the red one is 2, the second red strand is 3, and the fourth strand is blue black again. So we're going to be separating those 4, and we're going to be amplifying all 4. Okay, so the video that's coming up next, I just want to really emphasize the fact that we are splitting the denatured DNA. Oh my gosh, my hands are now. So we are splitting the DNA, and as you can see in step 2, we have done that by denaturing. We've heated it up. Now we've added primers. So the reverse primer is being added on it's going to the left side. The forward primer is going to the right side, and they're both starting from the gene of interest. Now we have elongated the DNA, as you can see in step 4, and then in step 5, as I've said again, we are splitting these 2 strands apart because we're going to be amplifying the red stuff and the blue stuff again. Okay, so that was the first cycle of PCR. Now we're going to move into the second cycle, or second and third cycle, I guess. So now we have denatured our DNA, and so or DNA slash primer A, I guess. We're gonna call it the DNA primer hybrid, but we all know that it's our, you know. So we have taken our gene of interest, we have added our primers, which are in red, and now we have split them apart. So because of that, we are going to be adding primers to the gene of interest and the red primer for both sides, okay? I've been trying to film the rest of the audio and I keep messing up. So we're gonna get this right, okay? Once and for all. We have added our green primers to both strands, both the red elongated primer strand and our gene of interest strand, okay? And we're doing this twice because there are 2 sections that we have just split up, or I guess the 4 strands that we have just split up from the first cycle. We are adding our green primer, our forward primer, to the bottom one. So the red strand that we have, we are adding the green primer to the red strand. The green primer goes from 5' to 3', and the red strand goes from 3' to 5', okay? We are adding the reverse primer to the blue section, the gene of interest section, starting from 5' to 3', the reverse side. We don't want the rest of the sequence that's in black because we only want the blue sequence. That's our gene of interest, okay? So now that we have done that, we are going to elongate by doing this with nucleotides and DNA polymerase once again. That is step 6. Well, I think it should be step 7. I did not write the steps, but step 6, we've added the green primers. Oh, I just changed the time lapse. Okay, so that has been elongated. That is our second cycle. We have done a third cycle in the bottom one. I did not do the primers again. I did not feel like doing the primers. But we have split both the green from the blue and the green from the red. So we are adding pink to it instead. Okay, so what we have done here is we have added pink primer to both the blue side, or the blue section, and the green section, and then another green section, and then the red section, okay? And so I'm going to break this down in another like small clip so I can show you. Okay, so we're going to go through this one more time, a very quick recap. We have split apart, we have denatured our red strand from our gene of interest. We are going to add reverse and forward and reverse primers, which is our green in this case, to the strands. We are going to elongate them with our nucleotides, our DNTPs, and DNA polymerase to get another set of strands. The third cycle, we are going to denature all of these strands. So the green will be split apart from the blue, and the red will be split apart from the green, and we're going to add another set of primers, which in this case will be pink. So we're going to elongate those primers, and we are going to get this whole set right over here. Okay, so now let's move on to primer design and in-frame importance, but I'm also going to touch on restriction sites. because this is important. When you are doing your primer, you're going to want to attach restriction sites for the amplified DNA because you're going to want to be cutting them and inserting these gene of interest into your vectors. So I'm going to do a little bit of an overview of primer design, and then we're going to go into forward and reverse because they're two different primer designs. So here I have a vector, and there are different restriction sites or enzyme sites that the endonuclease is going to cut. And so you're going to want to implement those into your vector. And because when you want to implement those, you can put your gene of interest, you can cut it and then put it in your vector. So it's perfect. So now we have our sequence that we want to clone, and I put little green markers there because I'm going to split them. Because you're going to want to put 21 base pairs of your gene of interest into 21 to 25 into your primer. So to do that, I just split it in half, and I have also divided them into triplets because this is important when you're going to be in frame. If you're out of frame by one or two nucleotides, then what's going to happen is that it's going to shift over by one or two or something like that, and the protein's not going to be properly transcribed, and so we don't want that to happen. So it's very important to be in frame when you are doing primer design. Okay, so moving on into designing a forward primer, what you're going to need is you're going to need a restriction enzyme site code, okay? And I'm just going to take the enzyme or the endonuclease enzyme BAMH1, and you can use any of the enzymes, but I just decided to use this one. The code for this one is GGA-TCC. That is the restriction enzyme site that is going to be cutting. That's what the endonuclease is going to cut for that sequence. And you're also going to add a couple of bases as a buffer. And so the one that is in red, the GC, is the buffer that I've chosen. You're going to want to choose G's or C's or just do GG, CCC, just any G or C, or you could just do straight G's, straight C's. And this is because G's and C's are harder to break because of the fact that they have an extra hydrogen bond. And so it's also to prevent the endonuclease from cutting too far into, like, it could actually not even cut the sequence, it could just cut your gene of interest. So that's what you don't want. The next thing that you're going to do is you're going to want to add 21 base pairs, and that is just your gene of interest. So where I've split it in the earlier in the video where that little green marker is, that one is where I've basically cut off. You're just going to want to take 21 to 25 base pairs, and that is going to be your forward primer. So you're going to put together your buffer, your restriction enzyme site sequence, or that recognition site so that the endonuclease knows where to cut, and then your 21 base pairs, and that is basically your forward primer. So the next primer we're going to tackle is the reverse primer. Actually, I just inserted the forward primer photo for you in case you need to see the whole thing. But the reverse primer is a little bit tricky because of the fact that you're going to have to read it in complementary and then flip it. Okay, so for this sequence, we're going to take the last 7 multiples, or the last 7 triplets, or 7 times triplets. And so whatever is from the 3' end um, going to the left, that is what we're going to take up to the green side. And you just want to use your 21 base pairs like before, like I said. But for this reverse primer, what you need to do, actually, I wrote it wrong on the flip sequence of interest. You actually have to do the complementary sequence, or the complementary gene of interest, and then flip it. So what you're going to do is you're going to write the complementary sequence. What I recommend doing is when you have your gene of interest, write the complementary sequence, the whole thing. You're going to take that complementary sequence and you're going to read it in the reverse direction. So what I have here is it would be like TA, or it would be TAT, and then CGT. And so I kind of have that there on the bottom, as you can see with my fingers. But once you write out that complementary sequence, you're going to flip it and read it in the reverse direction. So as I have TAT and then it goes from 3' to 5', it's going to start from 5' TTA. As you can see on my screen, I have ATT. When I go down, I'm going to move the camera down in just a little bit. It's going to, yeah, so TTA. So you're basically just going to flip the entire sequence. And I'm only doing it up to the 21 base pairs. I'm not going to do the whole thing. But what I did is I did the complementary sequence of the gene of interest, and then I took only the section that I needed and flipped it. And that's what you're going to do. And like the forward primer, we are going to find the restriction enzyme site that we're going to use, and I used EcoR1. The code for that one is GATTC. So you're going to add it right in front of the flipped one. You don't even have to flip the restriction enzyme site. You don't have to flip it around like you did with the gene of interest, but you do need to have your buffer, which is the GCs, because remember, we don't want our endonuclease to cut the our gene of interest, right? Because we want to clone that or PCR it. And so yeah, that's pretty much what the reverse primer is. And now when we put this in our vector, what is going to happen is the endonuclease is going to cut that restriction enzyme site, or EcoR1 is going to cut GATTC, that recognition sequence, give it a little bit of overhang, like I talked about in the restriction mapping video. And if you haven't seen it, please watch it because I feel like it is a little bit important. But it is going to leave a little bit of overhangs, and that will fit into our vector, which will also be complementary with the sticky ends as well. So that's pretty much it for the reverse primer. It's a little bit more tricky than the forward, but it's not as bad. Okay, so next up we're going to be talking about the melting and annealing temperature. And they're going to be different for both the primers, so it's okay if you get two different numbers. The formula is as follows: melting temperature is equal to 2 times the number of adenosine plus thymine plus 4 times the number of guanine plus cytosine. You're going to add all of those up using the primers that you have. I just used it from the last example. And the melting temperature for my forward primer is 90 degrees Celsius. I don't know how it is in Fahrenheit, so do not ask me the Fahrenheit stuff. Only use Celsius. For my case, I've only used it. Reverse primer, same thing. I've gotten 84 degrees Celsius for my reverse primer. So they are both different for the forward and reverse, so that's okay. They are going to be similar in range, so that's fine. Annealing temperature is melting temperature minus 5 degrees. And because we have two different ones, you're going to go with the lowest temperature. And this is because it requires less energy. So that's pretty much it. We're going to go with 84 minus 5. We get 79 degrees Celsius for our annealing temperature. Next, we're going to be talking about quantitative PCR, which can be used to determine relative transcription levels under specific conditions, but it monitors amplification of a target DNA molecule during PCR. And so I have here a picture. I was going to put in a video, but then my mom came in the room and I was not able to take a video. But what happens here is that it's basically with fluorescence. The fluorophore signal is quenched, meaning that it is suppressed. It basically forms a hairpin, and because it's in close proximity, the quenching molecule basically says to the fluorophore, hey, you're not going to do this here. I don't want you to do that. But as it approaches the target DNA, um, it will basically kind of like open up and it binds to the DNA. And the fluorophore is successfully separated from the quenching molecule, allowing the fluorophore to resume and intensify. Okay, so what basically happens here is that the fluorophore or not admits, it's emitting a signal when there is a PCR product that is detected. And so if the gene of interest or your PCR product is at higher levels, then it will reach a predetermined threshold faster, and that will determine your relative transcription levels under specific conditions. Okay, so reverse transcriptase, which is our last thing we're going to be talking about, is used to determine RNA sequences derived from living cells, and mRNA sequences can be amplified in PCR. So to do that, we are going to be taking our mRNA sequence and we're going to be annealing a primer, or I guess the reverse primer in this case, and reverse transcriptase will be performed. So DNTPs will be added, and we're going to use DNA polymerase to continue the sequence. And so the primer will be annealed to the mRNA through 3' hydroxyl, that free end. And so we're going to get a creation of an mRNA DNA hybrid. And so what's going to happen next is we're going to denature our hybrid. And so we're going to take that little DNA strip that we've just formed and we're going to use it. And before we do that, we do need to denature it. So we're going to heat it up, all that kind of jazz. We're going to take our DNA part that we've done from the hybrid, and we're going to anneal a forward primer. And so this forward primer has a re- um, restriction enzyme site attached to it. Same with the earlier one, I just didn't mention it, but it's there. I have it in purple, and then the other one is in pink. But this one, the pink primer, the forward primer, has an RE site attached to it, and for an example, I put BAM H1, it could be like Hind3, Eco R1, all that kind of stuff, but it's going to initiate second strand cDNA synthesis. Now, after we have annealed the primer, we're going to continue elongating it like we had done with the previous step, and we're going to continue PCR with the same two primers, and you're going to end up with two strands of our DNA. It's not going to be an mRNA DNA hybrid anymore because we've already done that DNA stuff, and we're going to have restriction enzyme sites on both ends of the DNA. We're going to cut the DNA at their specific sites, and we're going to fit them into a suitable vector. The OG vector is in black, and it's fit into with complementary sticky ends for both the DNA and the vector. And that was all things PCR. So thank you so much for watching. I hope you enjoyed or have learned something, and let me know if there's anything that I should add. But I will see you guys in the next video. Bye bye.